Our final talk of today is going to be by Caterina Gratton from the University of Illinois, who's going to be talking about reliability and precision in MRI.
Thank you. I am very aware that you've been sitting through a lot of talks, and I'm going to be the last one right before your reception. So please bear with me, but hopefully there'll be interesting themes that I really think will connect with some of Russ's and Jack's presentations from earlier. I also am totally okay with questions throughout the talk, and maybe that will help you all stay awake at this late hour. I have a bunch of slides, but it's not like critical that I get through all of them, and I'd rather have a discussion. So please feel free to raise your hands or speak up if you're coming through the owl, because I won't be able to tell, but maybe Ariel can help mediate. Yes, great. So the talk today, I think originally I had given a title that was something about functional connectivity and two roads, and I am still going to talk about the sort of what my perspective is on two roads that are kind of taking dominance right now in the field, at least of large-scale brain networks with fMRI. But my focus is going to be a little bit different from the talks this morning, although both of them touched on this. And I'm going to talk a little bit about what the value is and how do we approach smaller scale studies rather than really big sample studies. And so let me just start off with a few motivating slides, maybe. Okay. So let me just start off with a few motivating slides. I think many of us.
Can you click on the page?
Yeah, maybe I can. There we go. So many of us have come here today with some interest in connecting different aspects of brain structure and function with behavior and cognition. And so we want to understand, how does the brain give rise to the complexities of our human interactions? I'll just tell you that from my particular interest in our lab is particularly focused on different aspects of goal-directed functions or control, sometimes called executive function. And so if I were to place a cappuccino cup image in front of you, you may have different reactions depending on your goals at a particular moment in time. You might drink that cappuccino cup if you just bought it and you were really sleep deprived, like some of us are, because we got flight delays yesterday. You might choose not to drink that cup of coffee because it's actually somebody else's cup of coffee, and that would be really socially rude. Or you may use that as a memory prompt to say, remember the last time you had coffee with a sibling. And so we're really interested in this ability that we have to very flexibly change our thoughts depending on our goals. And because we study humans, we have insight into different scales at which we can study this question. Can obviously, with different techniques, go all the way down to the molecular scale, but with non-invasive human neuroimaging techniques like functional MRI, we usually have access to spatial scales of about 1 millimeter or larger. And one of the things that has really emerged in the last 100 years or so of neuroscience is that when we think about these complex functions like goal-directed control, we often want to focus not just on one region of the brain, but how many different regions work together as part of these large-scale systems. And so with fMRI, we often study this through a technique called functional connectivity. You'll bear with me. I know many of you are probably experts in this technique, but just in case some of you aren't, or even for the experts in the room, let me explain how we do go about this in the lab so you can understand the images that come later and ask me more in-depth questions as you want. So with functional connectivity, sounds like a fancy term, but really all we mean is that we're taking some measure of association in the activity patterns between different brain regions with fMRI. So for example, we might measure the bold activity with fMRI in left and right motor cortex over time. And if I were to plot those time series, you would see they're very highly correlated with one another. You could also take coherence measures or other types of measures to look at the associations between their activity. But a region that's part of a different functional system, like a visual region, might show a distinct pattern with much lower correlations to those motor-related regions. Now, this has been known now for about 30 years. And maybe what was surprising when the studies were first done by Biswal and colleagues is that this pattern of high correlations between left and right motor cortex is present even when someone is sitting as still as possible, staring at a fixation cross inside the scanner. The spontaneous activity in the brain, these correlation patterns mimic the patterns of activations that we might see in evoked related task context. And of course, nowadays we can make these measurements all over the brain at once and represent the complexity of this data in the form of a correlation or association matrix, as has already been touched on in earlier talks today, where any given cell in this matrix just represents the correlation between a pair of brain regions. When I present it to you like this, you might look at that and say, wow, that's a lot of stuff going on. There's a noisy cloud here. What am I supposed to look at? Well, one of the major choices that comes up when you first present your data, and data visualization is very important. I think this is going to be a topic of one of your talks later this week. But in the form of correlation matrices, it's really important how you decide to order your different regions. And so if I place the regions in an order where I try to place regions that are part of the same system close to one another, you see this really nice structure emerge in this graph, where you can see these high correlations along the diagonal in this matrix and low correlations in the off diagonals. Now, because of this, we can really effectively use different data-driven clustering algorithms to identify systems of the human brain. We're using an algorithm called InfoMap here, but you could use other ones as well. And we can also then paint the brain in different colors depending on the network that they're part of. So when I show you these brains with all these pretty colors, what we've done is all these steps going up to this, where we've taken the spontaneous correlation patterns, we've used these data-driven clustering algorithms, and we've used them to derive these network maps. And I think that's pretty cool that we can do this in this sort of very bottom-up way of reconstructing systems of the human brain, even though with sort of a lot of layers of complexity. And it already by itself, you can tell that it's recapitulating some of what we already knew to exist in terms of human brain organization, like the presence of the visual system, presence of motor-related systems. You can point to them, the teal and the orange. Maybe go back. And as well as higher level systems like the yellow frontal parietal system, the red default mode system, and so forth. So this is pretty neat. And even though it seems very unconstrained, I'll argue to you that actually these group average maps have been shown repeatedly again and again across different studies to produce relatively similar answers to one another. So on the left-hand side, I'm showing you a map that was created when we took 120 people at WashU, stuck them in the scanner, took out their data, averaged their correlation matrices, and created a network map from those people. On the right-hand side of the screen, you can see the same thing that was done at Harvard. And so these two maps, even though they were done on different people with different scanners and actually using different clustering techniques, still show a lot of similarity to one another in the large-scale networks that were found. And so I think this was really exciting. This work, when it was coming out, led me to have a lot of excitement. I think others in the field about how we can use these techniques to address a lot of fundamental questions we might have about cognitive or systems neuroscience. How these brain systems, for example, might respond when someone has different forms of brain damage or disease processes. How they may differ across people and perhaps link to behavioral traits that those folks may have. How they may change over the course of the lifespan, and how these networks may be involved when we have different cognitive demands or different sort of contexts that we place people into. However, a lot of these questions, I'll note, really rely on us having good measurements of these networks, not just for a group of people on average, but actually for individual people. So we know that how their particular brain is responding, say, in a particular disease state. And this is challenging. This is something that the field has grappled with. There have been a number of papers that have pointed out that the retest reliability of resting state fMRI can be really low in most large studies that have been there. This is a paper from Stephanie Noble, who I think is going to be an instructor later this week. You can certainly ask her more about this. She's done a ton of work on reliability within people. You've already heard quite a bit about this paper from Scott Merrick and about the need for really large sample sizes to detect the small effects that we're getting with resting state fMRI. And lest you think this is only about resting state fMRI, let me just say that similar sort of discussions have been surrounding other types of measurements, for example, task fMRI, which has also been shown to have fairly low retest reliability. And so this leads sometimes to headlines like this one, which I think would be really discouraging to many of us in the field, saying studies of brain activity aren't as useful as scientists thought. So we could just give up at this point, but of course, I think as the other talks have pointed out today, there may be solutions to these issues, and hopefully you all are here to try to learn about those solutions. And when I think about these changes that have happened in the field since these papers have come out, I think about two major sort of strategies that are being taken to try to address this reliability or reproducibility crisis that we have in fMRI. And so the first has already been talked about quite a bit today, is this idea that we should collect larger sample sizes to get better powered studies and a better representation of the overarching population. And of course, these studies have been incredibly useful in terms of getting us to understand population tendencies and our ability to reproducibly detect even small effect sizes in brain behavior relationships. But I'll argue that there is another strategy that you can also take, which takes sort of a different approach where you try to maximize a signal and minimize the noise within your data so that you can detect some of these effects even with smaller sample sizes. And part of how we were thinking about this is that when the Merrick paper first came out, so we sat around and thought about what are the effects that have been most reproducible so far in fMRI. Some of these may have come from large sample studies, but actually quite a few of them have come from much smaller scale studies. It's interesting Jack used this as an example as well, maybe making a slightly different point. But if we go back to many of the original studies in, say, visual neuroscience, a lot of those were done with very small samples of people, around 10 participants in these studies, and yet they have reproduced across many other studies in the field. And so why did they work? I mean, partly maybe they're looking for very large effects that you can find in those early studies, but I think partly they're taking a strategy where they're looking for this effect within each individual participant and using the participants as sort of repeated experiments to show that you can find this effect again and again. And so power matters here, but the power that matters is not only the number of participants that you have, but also the quality and the amount of data that you have per person. And these sorts of studies are really well suited for looking at within subject effects rather than trying to group effects and sort of generalize to the population. And so let me make this a little bit, at their extreme, let me just point out that we're going to call this precision fMRI in the talk. That's a moniker that's been given to it recently. It's not the only moniker. Sometimes people call this deep fMRI or high sampling fMRI, or just this is how we've been doing it all the time in vision neuroscience. Why are you giving it a new name? Depending on the tradition that you come from. But let me make this a little bit more concrete. So this is a really nice graph that comes from a paper by Thomas Solaris and Kendra Kay, who is another instructor that you will be having later this week, where it thinks about how we might design neuroimaging studies. And it thinks about this along two major axes. The y-axis here being the number of participants that we have in a given study, and the x-axis here being the hours of fMRI data that we collect within a given study. And of course, in the ideal world, we collect lots of participants and lots of hours of data from everyone, but we can't do that because the NIH won't give us enough money and people won't come in that many times. So we're forced to make select options along these two dimensions. And so traditional fMRI studies often collected something like an hour's worth of data from 20 to 30 participants and maybe averaged all that data together, created group average maps, kind of like the map I showed you before. In resting state fMRI data, actually the tradition has been to collect something like 5 to 10 minutes of data rather than a whole hour's worth of data. So the problem is even more exacerbated than what I'm showing you on this graph. And so the big sort of N approach has been to increase the sample size relative to the small amount of data we collect per participant. So starting with the Human Connectome project, collecting about 1,200 participants, and now the ABCD, UK Biobank, HBCD, and so forth, collecting data from tens of thousands of participants to increase our N. But another approach you could take, and I'll argue is useful as well, is to, rather than increasing the sample size, increase the amount of data that you're collecting per participant. And so this is what I'm going to call the deep or precision fMRI approach. And lest you think it's just about collecting more data, I really liked this recent paper from Lean Cooper's, also from Kendrick's lab, pointing out that what we really need is more intensive human neuroimaging from these studies, not just more precision human neuroimaging, in that we need to collect more data, but it's also important to design studies well, collect data across a number of different, say, conditions of interest or populations of interest, and that it's important how you analyze and think about your data afterwards and do quality control. It's not something I'm going to talk a ton about in my slides, but please ask me if you have questions. In resting state fMRI, there are certain things that have come up multiple times within our work that matter a lot. How you actually scan, what are the parameters that you're using, thinking about alignments and how to improve those if we're trying to get really precise measurements, thinking about how we denoise our data, something that functional connectivity really struggles with, and also thinking about our different approaches to network definition. And in case any of you are interested, this is maybe a theme that will come up at other points in the week. But I think one of the things that comes out is it's really important to look at your data in multiple different ways. And there's an old tutorial that I had in past years at NeuroHackademy that I gave that shows you a few different ways of looking at your data from the very pre-processing stage all the way up through the network and graph theory stage. And I think I can put out a link, or if Ariel has already put out a link, you can access that if you're interested. And then I'm not going to talk about it in a great deal of detail, but I think Jack did a really nice job discussing this issue that we also need to be able to share this data. They're smaller sample data sets. They're often collected with sort of particular questions in mind. But if we want to get at that generalizability question, we really want to be able to merge together different aspects of this. And so at least in some cases here in the talk, when I've remembered to, I've tried to put in like the open neuro links for these data sets so you could access them as well, perhaps use them for your hackathon projects. Okay, so in the talk today, I'm going to sort of jump through a few different points about precision fMRI, the first being why we would want to do precision fMRI in the first place, and then the last three sort of highlighting recent papers that I think have done a good job of showing the power of these techniques. And so let me just jump into the first one. So when I'm talking about these precision fMRI projects, two data sets in particular have been used quite a lot, so I'm just going to introduce them. The first has already been introduced by Jack, and actually surprisingly not by Russ, but it is his data set called the MyConnectome project. And so inspired by the questions that we had about reliability of these resting state fMRI measures, Russ sought to really maximize how much data we could get on a single individual. So he bravely went into the scanner twice a week over the course of the year, generating a very large amount of resting state data, as well as some task data, survey data, blood draws, and so forth. And so this data is openly available on Open Neuro. You can access it there, and sort of constituted the first example of trying to maximize or the resting state fMRI reliability within one person. Now, this data set then inspired many further projects, including the Midnight Scan Club project, which I've worked with quite a bit. This was spearheaded by Nico Dosenbach and Steve Nelson. And the idea being, why not have just one person? Let's have 10 people. And each of these 10 individuals came in for actually 12 MRI sessions, 10 of which were focused on functional measures, in which they did resting state and a number of other tasks. It was called the Midnight Scan Club. I'll just point out in case anyone is interested, because this data was collected all starting at midnight, potentially something of interest or a confound. It was done that way because the scanner was discounted 90%. And so that was the way they managed to get this project to fit into their budget. It's no longer the case that you can scan at midnight at WashU for 90% less. Okay, so we've collected a lot of data. Why do we want to collect all this data from individuals? Well, one of the things that this does is it improves our measurement of brain networks within those individuals, and particularly improves the reliability. So Russ touched on this before. By collecting more data, we're going to reduce our noise, our sampling variability, just a principle of statistics, and this is true and within subject data as well. And so nicely, as was nicely shown by Tim Lauman and Russ in one of the first papers on his data set, if you measure the connectome from Russ over the course of the year, and you take that data and you divide it into two independent split halves, you can produce two connectomes that actually look really similar to one another, not just in the general scale, but even in the details if you try to match between them. And if you can't tell by eye, let me just say quantitatively, this is true. So if you take the correlation between one half of the data and progressive amounts from the other half of the data, I can show you we can get above 0.95 correlation between the two halves. So this is really high for fMRI measures in general. I'll also point out, though, that this takes more data than we have typically collected in many resting state fMRI studies. So here, this first red line marks the 30-minute mark. The second one is 100 minutes of data. And so I'll just remind you that when I was saying before, most resting state fMRI studies start with 5 to 10 minutes of data to start with, and that's before you take away data because people have been moving inside the scan. Now, this is not the be-all, end-all measurement. This is only on a single participant, but this point has been replicated now across a number of different data sets, including the Midnight Scan Club data set, where we can see similar types of curves for functional connectivity as a whole. And you can also extend this to looking at other types of measures you might care about from functional connectomes. For example, the network assignment, global efficiency, modularity, and so forth. And in fact, this is an approach that I'd encourage you to do if you have a new measure of interest, to use one of these small scale data sets, to use them as kind of your gold standard. How much data in an ideal world would I need to really measure this particular quantity reliably, and then design your project with that idea in mind. And this is something that we frequently do within our own lab. These curves are going to be affected by a number of other variables, like the resolution at which you are collecting the data. I think we talked about, someone asked about 3T versus 7T. That's going to affect how these curves look. Also, the structures that you're interested in. Some structures have inherently lower signal to noise. So again, there's not going to be this magic number of just collect X amount of data. It's going to depend on your specific question. Now, I think Russ touched on this before as well, but reliability alone is not enough. We also need to care about validity. So how good are these individual measures at actually picking up these like relevant functional neuroanatomical structures of the brain? And so I'll argue to you that across a number of different measures, these approaches that collect a lot of data and pay attention to these individualized measures outperform more classic group studies. So just to give you a simple example, in the original Midnight Scan Club paper by Evan Gordon, he looked at the consistency between the functional connectivity measured at rest for the motor network, so here in the teal and the orange ones I was pointing out before, and the task activations in a motor task where people are asked to move their face or their hands. And hopefully what you can see is that the resting state networks are actually mapping on really nicely to the task activations that are measured independently. And this was true consistently across the participants in the Midnight Scan Club dataset. And if you tried to sort of cross this, like use the networks from subject 1 in the task data from subject 2, it doesn't work nearly as well. So it's significantly worse. Or if you use the group networks, it's also significantly worse. And so this has now been shown across a number of different contrasts that these approaches improve the match between task fMRI and resting state fMRI, as well as other forms of validation, such as improved alignment between resting state fMRI and structural measures. So now that we have this high quality data with higher reliability, and I'd argue also higher validity, we can start to look at these individual participant maps and ask ourselves, what do people look like relative to one another and the group average? And do this with a little bit more confidence that what we're measuring and the differences across people isn't just sampling variability or some aspect of noise in our measurements. And so I'm just going to cut to one of the papers at the end because it's important to contextualize the earlier effects. When we've done this sort of thing where we characterize individual differences, this is a project I did together with Ben Seitzman, who is a graduate student at WashU. We can go through the brain in, say, the participants from the Midnight Scan Club dataset, and we can look at how their functional networks look like, in this case, all the regions and how correlated they are with this one little white dot shown up here in subject 6 from the Midnight Scan Club. And look at how similar that pattern from subject 6 is to independent group average. And so in this case, you can see that the two are fairly similar to one another with high correlations in lateral prefrontal cortex. So if you take a spatial correlation of the similarity between these, you'd get a strong value. And that's the case for much of the brain. But there are, in the same location, another subject, subject 2 from the Midnight Scan Club dataset, you'll notice a very different pattern here with negative correlations now with lateral prefrontal cortex. So if you take the spatial correlation between these two, you'd actually find a much lower value, less than 0.3 correlated with the group. And so when we do this, your first question might be, is this just a function of noise in our fMRI measurement? And what we find is that with these precision data sets, we can show that these same regions occur again and again across different sessions from this participant. So it can lead us to have higher confidence that these individual differences are something that can be reliably measured in this person. Now, of course, across people, the locations of these regions, which we call network variants, vary to some degree, the size and so forth That's a topic for later in the talk. But let me just say that they don't occur randomly everywhere in the brain. They tend to occur in certain locations of the brain more than others. So if I overlap the regions of network variance in the Midnight Scan Club, you can see that they occur very strongly in lateral prefrontal cortex, as well as near the temporal parietal junction with some hemispheric asymmetries. I can talk more about those if anyone's interested. And this was true in 9 subjects, small sample, hard to say. But this is a case where we can take these techniques that we've now learned from these smaller sample data sets to a big data set like the Human Connectome project and say, can we see this again? Can we generalize this to a larger group? And in fact, we can see a very similar pattern in the Human Connectome project, which is nice not only because it's a bigger sample, but it was also collected with very different spatial and temporal resolution. So I'll come back to this point at the very end, but I just want to emphasize that just because we do one of these approaches in one study doesn't mean we can't take advantage of sort of the other road or the other extreme of this. Sometimes you gain a lot by wedding the two together. Yeah.
I was curious about how you picked that seed region. Was there like a theoretical reason, or did you find that via some exploratory analysis?
This one?
Yeah.
I don't think there was a theoretical reason. It just happened to look different in that one subject, but you find them all over the place. So if I had seeded any of these other ones in the black circle, you would have seen the same thing in this participant. Yeah, but I will talk a little bit about ones that are more theoretically motivated in the next section. I thought I saw another hand. Did anyone have other questions? This is a good moment to ask.
Could you talk about those hemispheric asymmetries?
Yeah. So this is a project that actually one of the graduate students in my lab, Deanna Perez, took up. When we looked at this, we were kind of surprised that there was like a lot more of these individual differences. It seemed like on the right side than on the left side, but it was a small sample. And then we reproduced that effect in the Human Connectome project. So we were convinced that maybe there was something more real going on. And so Deanna fairly carefully then characterized the hemispheric asymmetries in these individual differences and found that yes, people systematically have more individual differences in the right hemisphere than they do in the left. Of course, it's a matter of degree. You have them in the left hemisphere as well. They do often sort of border language-related regions. So we think this might be sort of an anchoring phenomenon where there can be less variability, basically, in the left hemisphere because of language. But it's a little hard, obviously, to demonstrate that. That's just a hypothesis. And they do relate to some degree to handedness. So she was able to show that right-handers and left-handers differ in this asymmetry. So I encourage you to look up the paper in Journal of Cognitive Neuroscience if you want to learn more.
Did you look at the twins in the Connectome project?
Ooh, not for this, but for a related project. I'll get to that later. Yeah. Yeah.
Their relation also with the variation of the superanatomy or the
So we've been sort of working on a project that's been taking a little while with Jacob Miller, who's now starting his faculty position at the University of Miami. He's done a lot of this work on tertiary sulci and thinking about finer scale anatomical variation. There's great work being done out there in the anatomy world. And I think there will be relationships. They've shown some relationships to resting state networks in general. We have not yet shown that with these precision measures. Great questions. And I guess the broader question maybe is that general variation in sort of cortical thickness and sulcal depth does not relate to these individual differences. So it's not easily explained by just a measurement in anatomy. I think it's going to be these sort of more fine scale associations.
specific subject that had different functional, like might have a variation of the supercharges.
Yeah, so it's not like as a general, they may have a variation in a X tertiary sulci, which causes a variant. But if you look at like the level of deformation across the cortex within that participant, doesn't correlate with their network variance on average, nor does their sulcal depth, nor does their cortical thickness. So that's where I think it's like specific variants will have specific relationships, most likely. Any other questions? Okay, so this was just a teaser for how we think about individual brains, just to give you a sense of which regions tend to show variation and to emphasize that people do show a lot of variation in their functional networks. And so given this observation that people have a lot of variation in their functional networks, it sort of has driven us to ask a couple of different types of questions within the lab. One of which is, what are the sources and consequences of these individual differences? I'm not actually going to talk about that very much today, but it's something we're quite interested in, so maybe in the question session later. But the other of which is, maybe counterintuitively, are there common properties of brain network organization that we can see now that we've addressed individual differences? So let me explain a little bit more about what I mean by that with an example. So if we turn to the dorsal anterior cingulate cortex, middle frontal cortex, depending on how you want to call it, has lots of different anatomical terms as well. This is maybe like one of the most argued about regions in the human brain, especially in the cognitive control world. Many different people have different theories about what this region is sensitive to. Is it pain? Is it ambiguity? Is it conflict? Is it motivation? And so forth. And if you do a neurosynth data analysis of all these terms, you're going to see that they all show some level of activation in this region, to some extent overlapping across these different terms. But what maybe isn't reflected here very clearly is that there's lots of networks in this region in the group average map. You can see it here. And this is even more complicated when we start to look at individual participant maps, where now what we see is that, yes, maybe people have kind of the same general networks. They all have the purple cingulopercular network there. They all have pieces of the default mode network there in red, but they're not in exactly the same place across people. And so when we look at these meta-analytic maps or group study maps, as we have often in the past, what we're probably doing is mixing apples and oranges. We're mixing together signals that are coming from different networks across different people. And so can we improve our understanding of what's happening in the brain if we don't do that, if we try to respect individual differences in functional neuroanatomy? And so that's what I want to talk about in the next section. And I'm actually going to do this with a brand new paper that we are hopefully have out on bioRxives like within the next hour or so. It's been sitting with them. That's coming from a graduate student, Zach Ladwig, in the lab. This isn't because it's the only paper that's done this. There's actually a lot of really cool papers in the field. I'll briefly talk about at the end, but I wanted to give you a more in-depth look into one project we've done in the lab so you can kind of understand how we go about thinking about precision fMRI studies. And so in this project, we were interested from a more theoretically motivated perspective in trying to understand the function of the lateral prefrontal cortex and its network organization, in part because we think of this region as being really important to goal-directed control, thanks to a long history of neuropsychology. Phineas Gage came up earlier, the first example of sort of telling us about the importance of these regions. And yet, one of the things that has been shown is that it's actually pretty hard to find a lot of functional organizational principles of the lateral prefrontal cortex. So many studies, when you look at the activations across very different types of tasks, find largely overlapping activity and kind of big blobs in the lateral prefrontal cortex, giving rise to these ideas that these regions may just serve these sort of flexible, multiple demand constructs that are just sort of adapting depending on what you need to do at a given moment in time. The network views maybe add a little bit more nuance to this. They show that there are different networks that appear in lateral prefrontal cortex, but this region is dominated by this very large yellow frontoparietal network in a lot of studies, and that's what people focus on. And there are some studies suggesting, oh, perhaps there is some more organization, but it might follow these sort of slowly changing gradients from rostral caudal or dorsal to ventral with different specializations in the patterns across these regions. And while those different reflections may be true, I'd like to point out that these regions that we think of as important to control based on task fMRI and network studies overlap a lot with regions that we know are showing a lot of variability in their functional neuroanatomy based on functional connectivity studies. And so I think we need to sort of think about this when we interpret the results of these past studies and take into account these individual differences, both to assess common as well as distinct properties of the lateral prefrontal cortex. And so we collected a new data set in the lab that we're calling the PAN data set, soon to be on Open Neuro. Again, spearheaded by graduates in the lab, Zach Ladwig. Also, special shout out to Nathan Labora, who collected a lot of this data, is in the audience today, so you can ask him all the details you want about it. And this data set is inspired by the Midnight Scan Club, another small scale data set, just 10 participants, so we're not going to overly generalize these results. But we have quite a lot of data from each participant, 7 to 10 fMRI sessions. And compared to the Midnight Scan Club, what's a little bit unique here is that we've collected data in a lot of different tasks, especially tasks that we think of as important for different aspects of control, like working memory, sustained attention, and interference. And we also collected tasks that probe other aspects of higher level function like language, theory of mind, and episodic projection that have also shown activations in the prefrontal cortex. Also, just make a point, I said 7 to 10 fMRI studies here, and one of the strategies that we used in this project, and we've been using in some other projects in the lab, is to actually, and I think Russ sort of mentioned this, is to collect a smaller set of participants, in our case, 2 participants first, with double the amount of data that we thought we would need, and to look at the test-retest reliability of those measurements to sort of understand what sort of data will we want to collect in other people, can we optimize these measurements for the rest of the study. And so just to show you here, we did that in the first 2 participants, and we looked at the network maps from those 2 participants. And hopefully you can see by going back and forth is that we can produce very reliable network maps within these participants with this amount of data. Yes, go ahead.
You have a study on the effect size variation of the across subjects, or like a set of tasks because that the underlying assumption here is that effect size is probably about the same
across people. Yeah, so the question was, do I have like a systematic study of the effect size variation? We haven't done exactly that. What we have done is, which not in this study, but in other studies, we create these sort of reliability curves, which I guess is kind of similar. Yeah. Let me escape. Hopefully this won't mess everything up for people. So when you create curves like this across people, hopefully everyone can still see that. We can check on like how variable the reliability is basically across people. So each of the lines over here is a different participant in the Midnight Scan Club data set. And so you can see they're not exactly the same. There is going to be some level of variation. And so hopefully this gets at the question of sort of like how much do you need to collect to get everybody above that level of reliability? And here we're really just targeting reliability for these measurements, not some measure of effect size. Does that answer your question? But it would be cool to think about how to do something more systematic as well. Okay. So yes, so in these 2 participants, we can show we can get really reliable network maps. This is similar in the amount of data that we have used in the past. So we were relatively confident we could do this because we've shown this amount of data can produce good network maps across people. But I'll point out that this data set was collected using multi-echo fMRI and very minimal smoothing so that we're able to pick up an even finer scale features of network maps than have been done in past studies. And so that was part of why we pushed the amount of data a little bit more here. What hasn't been done maybe as systematically is also look at task activations and their reliability. And so we collected, again, double the amount of data in these task activation maps. And here I'm just showing you the comparison between a high load and a low load in a visual working memory task. And you can see that we can produce, again, really reliable maps of these task activations within these participants that even reproduce these fine scale features within participants of these network activations. I can tell you it's above 0.8 correlated across the split halves for many different contrasts. Surprisingly similar, actually, across the different contrasts that we looked at with about 30 minutes of data. And you'll notice, perhaps, if you go back and forth, that also the subjects look pretty distinct from one another. So it's not just that they're reliable within themselves, but they're different from one another. So armed with this sort of data, we started this knowledge that we're getting reasonably reliable individual maps. We started to ask our question of what does an individual lateral prefrontal cortex look like relative to what we've seen in past group averages? So as I mentioned before, the group average from past work really has emphasized this large yellow frontal parietal network with some other networks represented above. And what we see when we look with an individual is something kind of similar. You still get the same general networks that are present, but you'll hopefully notice that individuals just look more complicated than the group. And in fact, what they do look like is more patchy, is how we're describing it, with a lot more little network pieces that are interleaved with one another. And we see this in the network assignments. You can also see it in the actual seed maps. So it's not just something about the network assignments making things look more different from one another. And so when we quantify these effects, what we end up finding is that when you look within the individual, the frontal parietal network is relatively smaller than you would have thought from past group average data. So the group average data was overrepresenting the frontal parietal network. There are a bunch of other network pieces that are represented. These include the default mode networks, as well as dorsal attention networks in these various spots. And there's also this just higher density of networks, especially association networks within lateral prefrontal cortex, relative to what you would see in the past. So just to give you an example, this is the average individual network density within the people in the 10 people in our data set relative to the group average representation of network density. And you can see even in the group average, you have a lot of networks in the lateral prefrontal cortex. It's a region that we think of as important in having multiple network representations, but this is just higher within individuals. And what you're particularly missing is this more rostral region. If I take the subtraction, it'll be more obvious, that has this sort of coming together of multiple network pieces that just doesn't seem to be well represented in the group average. And so what is that? What is that region actually doing? Well, one of the things that we see when we look within individuals is that there's some motifs that are actually shared across these 10 people that are being missed in the group average data. So there's this frontal region, this more rostral region of the cingulate opercular network in purple. In the more posterior regions of the frontal cortex, this is usually flanked by a piece of the dorsal attention and the language network across the different cingulate opercular regions. But it doesn't seem to be true in this more rostral region. But lo and behold, if we look within individuals, we actually see that that region is flanked by the dorsal attention and language network, just in slightly different locations across people. So it's getting missed in this group average representation map. There's one subject that only had one piece, but actually sort of inspired by this observation, we went and asked with the seed connectivity, can we find any evidence of a language connection there? And it does look like there is indeed some connectivity of the language region, even in that one subject where it doesn't come out in the network map. And so what this results in is that there's this frontal region of the cingulate opercular network. I didn't show you all the network maps, but here I'm sort of unveiling the connectivity of this rostral region that has a high density of networks. And it includes the language and the dorsal attention network, as well as the other association networks seem to all come together in this location, at least in these 10 participants. So this is all functional connectivity. You might be asking yourself, why should I care? What does this matter for actual task activations? And one of the things that we and many others have shown, as I've already mentioned, is that the task activations seem to map onto these network boundaries. And so reproducing effects from Lauren DiNicola and Ed Fedorenko and others, what we find, and Rodrigo Braga and others, what we find is that the networks tend to align with different types of task contrast. So for example, the theory of mind, a theory of mind task will particularly activate the default mode network B. An episodic projection task will particularly activate the default mode network A. And a language task will particularly activate the language network. Now, there I'm showing you just averages, but let me just show you data from 3 particular participants to convince you that these individual networks shown in the black boundaries really overlapping quite nicely with the task activations within them. And they're doing so much better than the group average networks are doing for those locations. And in fact, in some cases, the group networks are completely missing the activation that you would expect to see. And so what we're seeing is an advantage for these individual networks in capturing task-related regions. And I'll point out that although this is true throughout the brain, it's not specific to the lateral prefrontal cortex, there's an added benefit in lateral prefrontal cortex to these individual network maps. So you can see in these bars the lateral prefrontal cortex regions compared to non-lateral prefrontal cortex regions. Everywhere you get a benefit, so it's all above 0, but there's a bigger benefit from lateral prefrontal cortex, probably because of the added variability in this region. And then I just want to say that even though we're looking for commonalities, we do still see differences. With 10 subjects, it's a little hard to know exactly how common these are across people. So let me just report that they exist. We see these sort of idiosyncrasies, maps onto those network variants I was talking about earlier. So for example, in 4 of our subjects, we see this pattern where a piece of the language network seems to cut through what would typically be frontal parietal network here. We don't see it in every subject, but in these 4, there's a piece, the language network has this sort of more medial piece. Now, if I map the underlying functional connectivity of that region, you'll see that it maps onto the patterns that we're seeing with the network activations, again, showing it's not just something about the network mapping. And when we look at the task activations, what we see is that the language test does indeed really clearly activate these regions in these participants, and another test doesn't. So it's not something about any other task. So a spatial working memory test sort of skirts around these regions in these participants, activating only the frontal parietal network. So even the idiosyncrasies seem to tell us something about tasks. All right, I'll just close this section by making the observation that we also were really interested in control. And so we looked at the activations across these 8 other control tasks that we looked at. The situation here is actually a little bit more complicated. They don't fit squarely within a single network, look like those previous 3 tasks I showed you. You get activations that seem to activate multiple control networks like the frontal parietal, cingulopurcular, and dorsal attention networks, and actually often seem to lie kind of at the boundary between these different networks, perhaps giving a functional significance to for the need for lots of borders that we have in lateral prefrontal cortex. Okay, so that was an in-depth look at just one recent project from the lab, where I'm hopefully showing you some of the ways that you could use precision fMRI data here to look into more detail at the lateral prefrontal cortex, showing some evidence that it's relatively patchy within individuals compared to the group. It has a lot of these borders. That in the group data, we may actually be misrepresenting the underlying data over emphasizing the frontal parietal network. And that task activations do follow these network patterns. It's not just about resting state, but with control-related activations occurring particularly at the borders of these control networks. And so, as I said, we're not the only ones that are doing projects like this to try to use precision fMRI to improve our understanding of brain organization as a whole. I'll just very quickly say that there's some other great work that's been done, for example, showing that what we commonly treat as one common default mode network likely is 2 independent default mode networks with different types of function. This is work from Rodrigo Braga and colleagues. We can also do similar studies in non-cortical areas. So Scott done some work in the cerebellum, for example, showing a large representation of the frontal parietal network in this region. And Deanna Green has also done a fair amount of work in the thalamus and basal ganglia, showing evidence of regions that are both network specific and integrative across different networks. And then I'll just highlight this recent Nature paper emphasizing that these precision approaches can actually be used for sort of making fundamental new discoveries about neuroscience. So even though in most textbooks, at least the ones I've always learned from in cognitive neuroscience, we might think of the motor cortex as having this homunculus that gradually transitions across different body parts that we can move. When looked at within individuals, Evan Gordon noticed that there was this weird pattern where there were these sort of 3 regions that were interconnected with one another and not connected to the any one single motor body part, but rather with movements across the entire body. And so he's dubbed this now the SCAN network, the somatocognitive action network, and it's been now reproduced not only in small samples of participants, but even in very large data sets like the Human Connectome project. And it's a discovery that we perhaps would not have paid attention to without that sort of high precision individual level data. All righty, I'll take a quick pause here, see if you have questions on those. I'll promise the rest of it's not in as much detail, but I wanted to give you a sense for the types, how we do precision fMRI projects, I guess. All right, I'll continue on. So at the beginning, I posed to you the idea that we're really often interested in not only looking at brain organization by itself, but how it relates to behavior. And I want to highlight that you can even use these small scale studies to look at brain behavior relationships, but you might do it in a different way than the Merrick study. So in the Merrick study and other studies we've talked about today, often what we're doing is looking across subjects. So we're taking a bunch of people looking for individual variability in some behavioral trait and individual variability in some brain trait and trying to correlate or associate the 2 with one another. That precision fMRI is not going to shine with that. It doesn't have the power to do those kind of studies. But where it may shine is by looking at relationships within a given individual and trying to understand how that particular person's brain differs over time, for example, or in response to particular conditions. And so a few studies that I think have really highlighted the potential of precision fMRI. One is by Tim Laumann and Nico Dosenbach, looking at how the brain networks respond in the cases of injury, neonatal stroke, perinatal stroke. And another actually from Nico's group as well, looking at responses to major interventions. And then not exactly behavior, more brain-body relationships, but there's a really nice line of work from Emily Jacobs' lab looking at how brain and hormonal changes are correlated over time. And I will give you a flavor of these other studies because I think they're pretty cool. I think there's time, but feel free to interrupt me. So this study is actually it was spearheaded originally by Nico Dosenbach and then carried forward by Tim Lauman. And it was a case of a patient that was directed to Nico's practice. So he's a pediatric neurologist, and he had a patient come in, and he was referred because he played competitive baseball. And but his coach noticed that he had very weak right hand relative to left hand. He was a left-handed pitcher. And so he eventually made its way through, and they got an MRI, and they checked him. He's also worked as a mechanic in a shop, so you can see he's able to do pretty fine scale mechanical things. And yet when you scanned him, what you end up finding is that this kid is missing a very large amount of their cortex, including most of their motor cortex in both hemispheres. So it's pretty amazing to think that our brain can do this and sort of carry out motor abilities. He's also doing fine cognitively, similar to his siblings. They eventually traced it back that he had been ill right after birth, and so they think it was associated with a perinatal stroke that occurred at that time. And one of the things that occurred here is that they were able to then scan him a lot of times, just like they did in the Midnight Scan Club. This is actually the original inspiration, I think, for collecting the Midnight Scan Club to create a control group for him. And what they found is that if you looked at both the networks, which are the outlines in purple here, as well as the task activations in motor-related tasks. Why can I not find my cursor? There it is. You can see that he does have activations for left and right hand motor movement, but they're not in exactly the same place as they would be in other people. So for example, in the right hemisphere, they're really all localized within this very small area of remaining tissue that is present in the right hemisphere. And that is allowing him to be a competitive left-handed baseball player. In the left hemisphere, it's actually displaced. These motor activations are now occurring in the parietal lobe. Seemed like there was also some changes in white matter connectivity that mapped onto this. And so they're systematically posterior to where we would get it. And if we map the networks as a whole, you see that all the networks have sort of rearranged themselves around this new pattern that we're finding in this patient. So of course, this is by force, by obligation, has to be an N of 1 sample. You're not going to have a lot of patients with exactly this pattern of damage, but you can use these approaches to more reliably try to understand what's happening in this participant's brain. Another study that was carried out in Nico's lab by Dylan Newbold, who's a graduate student with him, was to look at how plastic are these network changes over time. And so they had 3 participants, 3 monkeys, as they like to call them in the study. They had them come in for 2 weeks of scanning every day, 2 weeks of scanning every day while they had a cast on their dominant arm, which is the right arm in this case, and 2 weeks of scanning afterwards. And so they looked at the connectivity of the left and right motor cortex. I use this example at the very beginning because it's like one of the easiest kind of patterns to see that the left and the right motor cortex are really highly correlated with one another. And what they saw is that that was true in each of their 3 participants. But then during the casting period, this connectivity dropped dramatically. And then after the casting period came back, and in fact, what you could do is in each participant, you could map the patterns of functional connectivity change within this, showing that you can get these changes in functional connectivity in responses to interventions. Laura Pritchett and Emily Jacobs have also shown that you can see changes in functional connectivity in response to changes in individual hormonal levels. So in their first study, 28andMe, they measured one participant's hormonal levels every day over the course of a natural menstrual cycle. Then they repeated it again on birth control. And they were able to track then patterns in functional connectivity that tracked with these changes in hormonal levels. And very excitingly, recently, they extended this work to not only look over the course of a menstrual cycle, but also to look at what happens in a woman's brain during pregnancy. And so they were able to scan this participant both before, during, and after pregnancy, and in conjunction with measuring their hormonal levels. And we're able to show these changes in structural connectivity, gray matter volume, cortical thickness, and so forth, some of which normalized after the pregnancy, some of which did not, emphasizing the important role that hormones may have on our brain. And so there are many other studies that I could perhaps talk about, but I think one of the exciting directions that has already been touched on is that these kind of precision studies can also be used to power many different types of questions, thanks to their availability and sort of the open data sets that are being put out there. And so I just want to mention a couple. So the first here is the individual brain charting data set. This is only 12 participants, but each of these 12 participants has data from 150 different task contrasts. So you could use this data to then examine the relationships across these task contrasts within a person. And then it's already been mentioned, but the natural scenes data set from Kendrick Kay's group has data from 8 participants. Each of them completed 40 hours of fMRI testing over the course of a year, while they watched more than 73,000 natural images. And so this data set in particular has been used to power a lot of more visual neuroscience studies into building better models of human vision. All right, and so maybe I'll take a quick pause just in case there are questions there. Otherwise, I'll get right into the last. Okay, the last point I wanted to make was that, sort of coming full circle, that we can also use these precision fMRI studies to improve our characterization of how brains differ. Now, of course, this needs to be done in combination with bigger data, and maybe it shows an example of how you might unite these two approaches of smaller sample data sets and bigger sample approaches to generalize our findings better. And so I introduced this idea that we can use precision data sets to find these places that are really reliably differ between an individual and the group average. And so this is something we've now done in other data sets like the Midnight Scan Club data set more systematically, arguing that when you look at this, these individual difference locations are really stable over time, looking a little bit like traits of the underlying brain. And more recently, we've also shown that they seem to come in different flavors from one another. And so this is a project carried out by research tech in my lab, Ali Duretsky, where she noticed that when you go through the brain, what you find is that some of these individual difference locations occur right at the borders of networks. So if you follow this sort of circle across the different plots, you'll notice that this location that looks different in subject 6 in the Midnight Scan Club is actually a place where the yellow territory, the frontal parietal network, seems to be taking over territory that would normally be part of the cingulopurcular network. And so we see examples of this, and maybe your intuition is this is a lot of the individual variability we have in the brain. Some regions are a bit larger within a person. We've known this for a long time. V1 differs quite a bit in size across people. It should be true of other regions too. One of the things that was maybe a little bit surprising when we started down this road is that that's not the only type of individual difference we see. We also have examples where there's this, say, little red piece of the default mode network hanging out in a place where there usually isn't any default mode network at all nearby. And so we call these ectopic intrusions. They're at a distance from the typical location for these individual differences. And so I didn't want to get into it too much because I want to leave time for questions, but across a series of analyses, Ali showed that both of these types of individual differences are quite common, both in the Midnight Scan Club dataset, as well as the Human Connectome project. They do differ in some of their properties, where they're likely to occur, which networks they connect with, which task types of activations. But sort of circling back to the question earlier, we also looked at this in the twins and the Human Connectome project. And what we were able to show is that both forms of individual differences do show heritability. So they're more similar in monozygotic twins than in dizygotic twins and siblings, than in unrelated individuals. But this pattern seems to sort of be emphasized or exacerbated in the border variants, suggesting that they're maybe more guided by genetic differences across individuals. So that was exciting to us. Also very exciting, if you've been following in the precision fMRI field, was this paper recently from Chuck Lynch and Connor Liston, where they were looking at individual differences in the context of individuals who had depression. And what they had done is they collected this sort of multiple session repeated scanning study of a very small sample, 3 individuals, a small set of individuals who with depression came in every month for many months. And this was very discovery science. They were mapping the networks within these individuals, and they noticed, it looks a little weird relative to other past publications on networks, where this black salience network looks a lot bigger in these participants than we have typically seen in the past. And maybe it's a function of just those 3 subjects that they collected. Obviously, we can't know, but what they then went out and did is they said, now that we have this thing that we can find really reproducibly again and again in these participants, maybe we can build a tool, a template to identify this in larger data sets where we have less data. And that's indeed what they did. They looked at this in these replication, much larger data sets, and found again the same pattern where the salience network seemed to be expanded within individuals with depression relative to healthy controls. Exciting for us, given our past work, is they also looked at this with reference to the border and ectopic variants and showed that most of these differences were due to a border-based expansion of these networks. And actually, they were able to show, you can see this even before the onset of depression in the ABCD data set, suggesting that it may serve as a risk factor of later development of depression in individuals. And for the most part, what I've told you so far is precision fMRI data sets done in young adults. And that's because that's most of what's been done so far in the precision fMRI literature. But we and others are trying to sort of push which groups are being included in precision fMRI studies. In our lab, we're doing some work going into healthy aging, so collecting these data sets across the course of the adult lifespan up to ages 75. Shout out to Jake, who is spearheading a project in the lab in collaboration with Megan Campbell at WashU to look at what these networks look like in individuals with Parkinson's disease, who also have a bunch of other measures, so we can look at their characteristics relative to differences in symptoms and so forth. And I also wanted to point out that other groups are extending this the other direction, going into kids. So this was just actually put out on bioRxives, a paper from Deanna Green's lab looking at precision fMRI within kids ages 8 to 11. The Damien Fair's lab has been extending these techniques to the ABCD project to map individual brain networks within that data set. And then Chuck Sylvester's lab has also been looking at what these brain networks look like in neonates. So I think these are all like, at least these first 2 papers came out just like this summer in the last few weeks. So I think there's a lot of exciting stuff that we will learn in the next few years based on these data sets. So hopefully this sort of round robin has given you a sense of what people are doing with precision fMRI right now. Of course, it's not enough to just say, I'm going to have a smaller sample of people here. You want to really emphasize and take care to think about the reliability and validity of your methods and how you could then go about generalizing it to larger samples. But I think there's a lot of potential, especially if we think about how to unite these 2 pathways moving forward. And if anyone's interested, there's a special issue that we put out a few years ago. Now there's, of course, new papers, but there's a bunch of great papers in this special issue that you can look up other folks that are and how they're thinking about these precision data sets. So I'll point you to that. And then I would like to thank all the people in my lab who've been carrying out these projects, as well as alums, some of which are in the audience, and then collaborators. A lot of these projects, much like the sort of big consortia projects, really take a team of people who are working on them just because they're kind of complicated to collect with all these different measures and the people coming back. So I definitely encourage you that this team science approach has been really beneficial in my work, and I think you would find the same. So I encourage you to consider that as well. And I'm happy to take any questions that you have.
Question from online. Would high precision MRI imaging also benefit in better characterizing subcortical network connectivity, or is it still better for cortical network characterization?
Yeah, that's a great question. I actually think in some ways you might get more benefit from subcortical network connectivity than in just cortical, because the smaller scale structures there. Basically what we see is the smaller, the finer scale structure that you want, or the finer scale detail you want, the more benefit you're getting from precision fMRI, because it allows you to account for individual variability that may sort of be too large at that point. And so we have done some work on this, and by we, I mean Deanna Green had this project that I mentioned earlier in looking at the thalamus and the basal ganglia. There's also been work from Randy Buckner's lab looking at those structures, cerebellum as well. that has done some really nice initial projects. Rodrigo Braga's lab is actually using the natural scenes data set to now extend this with the power of 7T and sort of the added signal that we can get there. So I think there'll be more to come as well. Yeah.
I have a question. So I was interested to see that you're starting to work on aging populations. There doesn't seem to be a lot of work on sort of these functional networks in aging. And I guess probably one of the reasons is that there's all these confounds. Aging people, their vascular systems also changing. So this is just more like a technical question, just because I'm interested in this.
Yeah.
What are you doing to deal with vascular changes?
Yeah, I think it's a hard question. There is, yes, sorry, I'm very bad about this. Repeating the question. So what, is there not a lot of work on functional networks in aging, perhaps because of the many confounds that happen with aging? And in particular, how do we think about vascular confounds and the changes in the cardiovascular structure? And so I don't think there's an easy answer to that. First, I would say that there is actually a fair amount of work that's done in functional networks in aging that I did not talk about today. But one of the effects, people have done these sort of larger scale studies. One of the effects that people find maybe most reproducibly is that functional networks seem to become what they've termed desegregated. So more overlapping in time with age. And a number of different groups have shown that. But I think you're right, and this is the way we're thinking about it, that it's a little hard to interpret those results for a number of reasons. One is motion. Older adults move more. Often we don't have that much data. Another is maybe there's individual variability. These approaches have always used these sort of group average network approaches, or almost always use that. And then vascular confounds are also really important. And I don't think there's an easy solution to that. One is to measure it. So we, for example, have been trying to collect things like MRAs and MRBs and measures of cardiorespiratory fitness in our adult lifespan participants. We also are starting some collaborations actually with others at the University of Illinois that are really interested in this interplay between cardiovascular changes and brain changes. They're not independent from one another, right? So if your vascular system breaks down, that usually does also cause damage to the underlying white matter and gray matter and neuronal activity. And that's been shown across a number of studies. In fact, is one of the leading hypotheses for one of the problems that happens in Alzheimer's disease. But I don't think, I guess besides measuring it, I don't have a perfect answer to your question right now. Yeah, go ahead.
I think of the methods that try to achieve the various techniques realign based on functional variation, like realign brains and maybe have the best of the two worlds in some sense. What's your take on those? Like, this all those techniques like multimodal transport or whatever, any of those things.
Yeah, so we use some of them in the lab. So there's, more, maybe not basic, but anatomy driven only techniques that will like free surfer as a basic level, and then sort of expansions of that have been driven by the Human Connectome project. Those by themselves don't erase the functional network differences, which
and with functional data.
Yeah, and then with the functional data, you'll have a variety of tools like MSHBM, which has been developed by Ruby Kong and Thomas Yo's lab. We're using that in some projects within the lab. And there, I think what we've tended to see, but we would really, and this maybe gets to a broader question, is that they work really well at giving you an individual network map within your participants. They don't always capture all the individual data differences that we see when we have very extended amounts of data, because they are a bit constrained purposely so by the group template. Of course, there may be improvements and sort of that we can do on these methods moving forward. In the lab, I sort of mentioned this beginning, one of the things that we're doing in a lot of projects moving forward that we've tried to do is maybe a little bit mini version of the multiverse, where there's not like one perfect way of defining networks. So we'll often use like a very data-driven method like InfoMap together with template matching or MSHBM, which has a little bit more priors built into it. Try to look for consistencies that we can see across these methods rather than trying to depend it too strongly on one of them. Also the seed maps that I showed you before, looking at the underlying data. But yeah, I think they're really useful. The other technique that I think is also quite exciting that we've only played around with a little bit is hyperalignment. So that's trying to go to fine scale features and trying to align across people, either based on like say movie watching or based on the functional connectivity itself. And so one of the things that's been shown with that is that you can improve, say, brain behavior prediction by using hyperalignment. Phelan has a really nice paper on this. And so I think that's another promising method that we want to implement as well. Yeah, go ahead.
Spend a little on testing how much data is needed for reliability in once you like that first second handful or full subjects that you put in extra data for.
Yeah, and just to be clear, you may not even need to collect your own subjects because so many of these data sets are available. You may be able to look at some of them that are on Open Neuro and just get a sense for it. But the approach we've taken in the lab, if there's like a new measure, or even if it's not a new measure, is that we try to ask ourselves, like, how accurate do we want this data to be? First off, if we're really looking at individual data, we really try to be above the 0.8, 0.85 mark on reliability. And so then what we do is we take the data, multiple session data, say from Russ Poldrack, you have 80 sessions, take 40 of the sessions, set them aside. We call those our gold standard, basically, derive our measure from that gold standard. And then we'll take progressive amounts of data from the other half in usually like 5-minute chunks, because that's about what we collect. And we say, how quickly does that data sort of reach this, how similar does it get with progressively more data? And of course, it'll get more and more similar as you have more. And where do we hit that sort of 0.85 mark? Maybe it's about 30 minutes. That's what we've seen for a lot of, say, cortical network connectivity. So in future studies, we don't need to collect 80 days worth of data on our participants. We can collect half an hour's worth of data, or maybe 40 minutes because we want to count for motion, but just 40 minutes worth of data in our participants and be pretty confident that we're getting reliable data there. So that's the general approach. Of course, this will differ a bit. So for example, in the subcortical regions that were asked about earlier, you probably need more data than that. That's what the initial studies have shown. You might need more like 100 minutes of data than 30 minutes of data to get good test-retest reliability of that 0.85 mark. So that's where I'm saying there's not going to be like one sweet spot answer necessarily, but you can use these other data sets to look at the measurements in more detail. Yes.
So a lot of these regions are, they go through drastic changes to later adolescence and young adults. And this made me think about like different networks in brain and fighting for your computing for the limited resources, limited real estate. And I'm gonna ask a big question. Have you seen any evidence for like maybe certain two networks going through like they go through similar changes, or like
that they compete with one another or something?
Maybe network A becomes more dorsal and involved, network B becomes more rostral, something like that.
Yeah, and I've been bad about repeating it, but let me try to restate your question. You can tell me if I have it right. So your point is that these, there are many changes that happen over the course of development and aging in the brain, and are all the networks kind of going willy-nilly? Are there certain pairs of networks in particular that maybe trade off with one another, have particular relationships with one another? And the answer is yes. So I didn't emphasize this, but for example, in the network variance literature, we do see it's not that like the frontal parietal network will become any other network often in people. When you have a variant in, say, the frontal parietal network, often will be either default mode or singular opercular, say. So other control maybe nearby networks depending on the place. And you could maybe imagine experience-dependent plasticity sort of driving that, where you have certain types of inputs at certain moments of development that would promote then a region to be expanded in one network relative to another. So I definitely think we see examples of that. And there are also clear anatomical relationships, relative anatomical relationships, not absolute ones, but that are preserved. And so Rodrigo Braga has actually documented this quite nicely for the two default mode networks. And I'm going to forget the details of them, but if you look at the two default mode networks, they often lie kind of next to one another. They seem to have these independent responses. They're not that highly functionally connected to one another. They respond to different tasks. But depending on the zone of the brain that you're looking at, they'll have conserved relative relationships. So one will always be more dorsal, one will always be more ventral, one will be more caudal, will be more rostral. And we kind of see that in some of the lateral PFC locations as well. And actually, Rod has really nicely written this up in an annual review paper that we should have coming out later this year. So I'd encourage you to look that up if you want to see a description of that as well. Yeah. I thought I saw a hand over there. Yes.
Work on like localized treatments for like psychiatric disorders, like the individual, like the patchiness of the LPC and individual network, like does that make people more or less receptive to like neuromodulation protocol?
Yeah, I mean, the patchiness, it's hard to say. We've only done this in 10 participants so far, but we are thinking along those lines. So let me, maybe two pieces of data. So one is if we think about, say, TMS protocols, it's a lot of those are targeting the lateral PFC and sort of those regions not too far maybe from that location I was showing you with the singular network and all those networks coming together. And so on the one hand, you could think maybe that's good. You want to TMS a region that has lots, you might hit lots of different regions, and that's exactly what you want to do in your protocol. Maybe that's not what you want to do, and you really want to target one specific network, and that might be hard to do, honestly, in the lateral PFC based on the integration we see. But what I will say is that there are a number of papers that have now come out from various different groups, not that I've been involved in, but that have shown that if you use individualized measures to identify your TMS target, that you improve outcomes, or at least there's a bunch of retrospective studies showing that it can help to predict some of the differences in outcomes shown with TMS in depression. And then one prospective clinical trial study, I think called the SAINT study out of Stanford, showing that it can improve outcomes of individuals with depression in terms of targeting. So definitely in terms of neurostimulation, I think these individualized approaches have a lot of potential. There's also another study that I didn't talk about because I felt like I was talking too much, but it's a kind of a cool study from Josh Siegel. Did I take the slides out? Might have taken the slides out. So Josh Siegel is the head author, and he was interested, not in a clinical population, but just trying to understand the effects of drugs on brain networks. And so he did this study where people were repeatedly scanned when they were under the influence of methylphenidate or psilocybin. And what he was able to show is that with methylphenidate, you had a little bit of change in brain networks. It wasn't huge, but there was like this consistent change while under the influence. With psilocybin, there were really dramatic changes in the synchronicity of functional networks. They went away after people, the next day after the effect wore off. For the most part, there were some smaller scale changes with the hippocampus that seemed to be consistent over time. But there do seem to be these sort of effects that you can track within individuals using these network approaches. And so that was a Nature paper also like a year ago, I don't know.
I have a question, when it comes to like observing certain like showing small effects in the data sets, we have a lot of data from participants, and you were able in one case to then show the same result in the human connection project, where I'm assuming you have a lot less data. Can you normally make this jump to the bigger data sets, or is the fact that they're really using a lot less resting state data a big limitation?
Yeah, I think it depends. The Human Connectome project actually has a fair amount of data for a big project. So it has 60 minutes worth of data per participant. So that's an easier jump, I think, to make, because as I had talked about, some of these measures do become pretty reliable with about 30 minutes worth of data. UK Biobank, I think, has 6 minutes of resting state data per person. ABCD has 20 when you start, but then kids wiggle a lot, so you're often down to about 5 minutes of data per kid. So it can be more challenging in those data sets. That isn't to say it's not possible. The SCAN project with the motor interrupting regions were identified in the UK Biobank, because then I think we kind of had a good template. We knew what to look for. And so once you have a good template, you can design techniques to go after them. So I think that shows promise in how we could use similar methods to identify noisy signatures, even in these data sets with less data per person. But I don't know that there's always going to be this sort of perfect match. Oh, and I guess maybe one other strategy to consider in those data sets that I'll mention, it's been used a little bit in the ABCD, is that you don't have to use just resting state data. So in the ABCD, there's a bunch of task data. And so one strategy you could use is try to glue together the rest of the tasks to increase the amount of data you have. Of course, that might come with some state level changes based on the tasks, but we and others have shown that at least for these individual difference locations, they're relatively minor and a lot smaller than the, say, difference between having 5 minutes and 40 minutes of data. And so I think that's also another effective strategy that can be used. Yeah, go ahead.
Not with inside their TMS and how that changes the types of considerations of the methodology.
Yeah, so the question is about multimodality and if that changes the considerations and methodology, in particular fMRI with EEG or fMRI with TMS. And so I guess, in my view, like the principles are similar. Like if you're interested in within subject changes, you still want to have a reproducible effect that you can find within a person. Obviously, you are doing a very challenging thing there where you're trying to unite fMRI and EEG and fMRI and TMS, but those kind of data sets have been incredibly valuable for the field. So I think it's an enterprise worth pursuing. There's actually a really nice paper from Talyazuki and Laus who collected a concurrent fMRI EEG data set of sleep data in the scanner, and we're able to use that to build a machine learning classifier to classify different stages of sleep, then applied it to like these big data sets, showed a bunch of people are asleep during a lot of resting state studies. But it sort of emphasizes that you can, I think, do this sort of thing. You can do it well, but I think just like with straight fMRI, you're going to want to care about these data quality issues, and it's going to be even harder probably in those data sets. But we haven't done that personally in the lab. There's a colleague I have at University of Illinois, Sepida Setagiani, is doing some really nice fMRI EEG that we're hoping at some point we'll be able to collaborate on that. Did I see a hand in this direction? Is that just? Yes, go ahead.
And I kind of, so while reading that paper and doing some research on it, I was quite stuck with the question of, is SCAN part of a motor network, like some other motor network, or is it something that is quite separable network? How do you think about the network function network? I mean, but at its core, I think it's like you have very consistently strong functional connectivity between these nodes that belong to the system. I think that's like a defined network. I just wanted to ask you if you have any insights about how to function network, or if you have insights specifically to SCAN.
Yeah, so the question is about SCAN and more generally about defining functional networks and whether SCAN, for example, is differentiable from the other somatomotor networks. Do we think of it as a separate thing? And so for SCAN, I think it's in primary motor cortex. So I think in its essence, it's a motor network, and it certainly responds in terms of activation and in motor tasks, but not in this sort of way where it's specific to particular effectors or particular body parts. And so I think, and just like you said, in the principles of how we define networks from a complex systems or graph theory point of view, we think of networks as regions that have high connectivity to one another and relatively low connectivity to other regions. So in that sense, I think of SCAN as a network. Of course, you can think of networks at different spatial scales and how they interconnect, and SCAN connects strongly with also the cingulopercular network, which is not in primary motor cortex, and maybe gives hints as to its region or sort of its functions. And I know that's a lot of how Evan is thinking about it as he continues along those projects. We're also sort of thinking about it together jointly in the context of other projects within the lab, although we haven't worked very much with SCAN specifically. I don't know that I have like a perfect answer to your question. Like, yes, X is now a network because I have high within network and low between, because often when you look at higher resolution, less smooth data, you'll often find differentiation between networks that you couldn't see And in just the same principle as going from group to individual data, also going from say 3T to 7T data might allow us to see more distinctions between networks and finer scale networks that we couldn't see before. So I don't know that there's ever going to be like a perfect answer. There are these X12 networks of the brain, and no more and no less. But I think that they can help us guide us in terms of thinking about what are the relative associations across regions, what are the relative functions, do some tend to cluster together, and at least that's how we think about it in the lab. Yeah, go ahead.
Do you have any consideration for the sample size in high precision study? If you're planning to like compare it to global average data, like what do you think could be idiosyncratic between just like more real?
Yeah, so the question is, do I have any like recommendations on sample size with these precision studies, especially if I want to compare it to larger studies and try to understand idiosyncrasies? And I don't have a perfect answer on this one either. What I will say is when you go small, especially like 10 people or less, it's going to be hard to interpret the idiosyncrasies. You can say that they exist, and you might say that they're reliably there and within a person, this person, they're associated with something, but it's going to be hard to say much more about how common they are in the population and sort of whether they're related to these brain behavior associations. Like I showed you that one with the language network cutting across. We see it pretty commonly. There are a couple others we see commonly. I don't know how common they're going to be in larger data sets yet. I think we need to look there. But with 10 people, I do think already if you're seeing it consistently across all 10, it, at least for me, gives me pretty good confidence especially when I can reproducibly see it within that one person. I know it's not just, something wiggly in that person. Gives me confidence that it's a pretty real effect. And often when we've looked at those, we tend to see it again, like we'll reproduce that effect in the Midnight Scan Club after we've done it in our PAN data set, or look in the myconnectome and see it again. And so I think the advantage of these small scale data sets is it's a little easier to jump across them and not that hard to collect like one more person to do a replication. But you do have to be like honest with yourself, right? Like just like Russ was saying before, you have to set some data aside as your discovery sample where you're going to identify this thing, and then look at another sample that you collect afterwards for that replication if you really want to discover something that you think is real. And so that's the strategy that we have used of like, yes, this is cool. I'm sort of out in the world discovering new continents, but I want to then replicate this again.
Thanks. So this concludes the official capacity of the first day of NeuroHackademy. For joining us. We will be back here in the other auditorium tomorrow at 9 a.m. The same time we started today for another talk prior to a school direct. And then the rest of tomorrow will be tutorial classes, and we'll put a little more information in the Slack about that before we start. For everyone who's here in person, we have a reception outside. Is here. Yes. Yeah. Okay. There's food outside. So please join us for dinner. And for everyone else, we'll see you tomorrow.
Okay, go ahead and hang up.